Stanford University.
Okay, and is this audible?
It's really quiet.
Really quiet. Something's wrong with the sound system today. How's that? How's that? Placebo? Maybe that would have worked? Okay, well, I guess I will just have to shout out, but okay. Help? All right. Where am I clipping where this is falling off? Is that doing it?
That's fine.
Okay. Good. So let's see. That is an event you probably should check out. And I'm still irritated you didn't want to sign football for me, but nonetheless. Okay. So starting off, as the large numbers of empty seats imply, not only is Monday the midterm, but today is not a topic that will be covered on the midterm. So in case you want to flee right now, here's your chance. I won't be upset. But what we do here is look at our last topic of our buckets, in this case, our neurobiology one. And the whole premise for this one is to cover the most wonderful, interesting parts of the brain, which you're going to hear squat about when you wind up in medical school. Again, from the other day, what they will teach you about endlessly is stuff that goes wrong with your spinal cord, or stuff that goes wrong with parts of your brain that regulate your bladder and your balance and motoric stuff. How come? Because, yes, you can't hear a thing. No.
He's going to try to find the sound.
Okay, somebody's off to get sound, but in the meantime, I will pantomime bladder problems. So what you've got, I will try, maybe in the back, just start waving each time I start hypoventilating and run out of volume. But what you got here is not the part of the nervous system that's easy to find out about because it's right on the surface. Not the part that people focus on because there's a lot of spinal cord injuries and you can tell exactly which is what's wrong by which toe is no longer moving. This is instead focusing on the part of the brain most centrally involved in emotion, the limbic system. Now, obvious why that should be of interest to us, more so than spinal pathways controlling movement. And in terms of making sense of the limbic system, this is going to be the most horribly multisyllabic diagram-laden class of the entire class. But in terms of making sense of it, what we will see is there's some very, very logical things going on, very logical strategies. Okay, originally, the limbic system was not known as the limbic system, but instead had a name reflecting the fact that people started off studying it in rats. And you take a rat brain, which looks sort of kind of like this from the side, and you got your spinal cord and cerebellum and cortex, and what is this thing at the way front there? This is the olfactory bulb. This is the olfactory system of a rodent brain, and this is drawn exactly to scale, down to the millimeter. This is huge. This thing is huge, the olfactory bulb, and its projections are 40% of the brain of a rodent. And when people began to understand where the neurons in the olfactory bulb were sending their projections, it was an area of the brain, the underside of the brain, which people very quickly began to call this region the rhinencephalon, the nose brain, because obviously this is what was processing all this whomping great amount of olfactory information coming in. And this area that we now know and love as the limbic system at the time was called the rhinencephalon. This dominated early on with the neuroanatomists, which is to say the people who just sat there and said, gee whiz, that's an awfully large part of the brain there, and where does it send its projections? As people instead in the 30s and 40s began to understand the function of this part of the brain, what emerged was a very different viewpoint from a different school, which is it's doing all sorts of stuff with emotion. And this picture of this region of the brain as having that role, for reasons I don't completely understand, somehow the people who liked the "it's all about emotion" started calling it the limbic system, and thus you had one of the most central conflicts of the 20th century, which was whether this thing should be called the rhinoencephalon or the limbic system, and thousands of innocent people were slaughtered before it was all over with. Brother pitted against brother, savagery, and what we've got, much of Picasso's paintings were about some of the atrocities committed by the rhinoencephalon people. But what wound up ultimately happening was a completely logical resolution. Rhinoencephalon, limbic system, is it about odor? Is it about emotion? And all you have to do is think like an ethologist and you got your answer, which is if you're a rat, there's not an emotion on earth that's not intertwined with olfaction. The rat's emotional world is utterly driven by olfactory information, and thus for a rat, the rhinoencephalon, the nose brain, is the part of the brain getting all the information coming in that has any relevance to the good, interesting, emotional stuff going on in a rat's life. So this massive resolution, peace in their time, suddenly this recognition that, of course, a rhinoencephalon is going to be sending projections, is going to be all a part of the brain that's doing emotion for you. Now, of course, as well-trained ethologists, you should immediately be having a relevant thought already, which is, well, so what's up with the rhinoencephalon famed nose brain limbic system in a species that's not some smelly rodent thing? In a species, for example, that's paying attention to enormous amount of, say, auditory stuff. How about all those birds with the birdsong business going on, or auditory displays of territoriality or things of that sort? And you look in those brains, and the limbic system is not the rhinoencephalon in terms of what part of the brain is disproportionately enlarged and what part of the brain is sending projections into the limbic system. Look at a bird brain, and it is not a rhinoencephalon, it's a ear-encephalon or something or other, and you got to think like an ethologist. And suddenly you're in a world where something as bizarre as electric fish, who communicate socially through electricity, have electroreceptors, which are sending a lot of information to this part of the brain. It is not the nose brain, it is the, among other things, getting whatever sensory information is most pertinent to my emotional life part of the brain. So the limbic system, initially utterly viewed as intertwined with olfaction, now instead recognizing as good neuroethologists what it's really about. Okay, so as an introduction to it, we need to start off with a very simplified, quick overview of large parts of the nervous system. You got bits of this last week, but just to give an initial overview here, just to orient what's happening. And what we see here, and again, a very accurate, technically drawn diagram, you see the brain comes in 3 different pieces, and each one has a different color. And this reflects the work of a guy named Paul MacLean, one of the giants in the field, and everyone is required to say his name at this point. And this model that he came up with in the 1950s or so, the triune brain, the 3-layer model of what brain function is about. So what you have is the most central, ancient, archaic, most phylogenetically conserved, which is a fancy way of saying that all sorts of vertebrates out there, this part of the brain is basically the same. This is the hypothalamus talking to the pituitary. This is the pathways going from the hypothalamus down the spinal cord, areas called brain stem, midbrain. This is what MacLean called the reptilian part of the brain, reflecting the fact that our section here is a whole lot like the type you would find in a reptile. This is the ancient stuff that does the purely automatic regulatory things. You've got neurons here that have to tell you something about temperature regulation. Ooh, back to that neuroendocrinology stuff. Let's see, what's a hormone that's got something to do with metabolism, body temperature, thyroid hormone? So you remember the hypothalamus being part of that cascade. The hypothalamus has a relevant hormone for the pituitary, which then tells your thyroid gland, all of that. So how does the hypothalamus need to know what's going on? That same feedback information stuff from the other day, there's got to be neurons in the hypothalamus that can tell your temperature. And too cold, let's get some more thyroid hormone out. Great, that makes complete sense in terms of regulatory loops, totally boring. Okay, so this is a part of the brain that keeps your body temperature right. It keeps your blood glucose levels sort of modulated in some ways. It talks a lot to the pancreas. It does stuff like it's monitoring your blood pressure in various places. It's remembering that you're running hard, so let's tell the heart to beat a little bit faster. Completely boring stuff. Just robotic regulatory circuitry and loops there. So this is this ancient reptilian part of the brain. Totally boring part of the brain until you get something wrong with that part of the brain, and suddenly it seems mighty interesting. Remind me, how many of you were in BioCore? Okay, lots of folks. People in there will remember I told you about one of the all-time creepy diseases that you do not want to get, which is called Odine's curse, which is where you get some lesion, some stroke damage to one of these midbrain areas. This part of the brain that does something very boring until it doesn't do it for you anymore. Odine was apparently some Greek nymph or something that Zeus was hitting on unsuccessfully at some point, and greatly frustrated, he put this Olympian curse on her, which was that she had Odine's curse, or maybe Odine's curse was what she had to say about him in return. But what this was about is she lost the capacity to do automatic breathing. Whoa, that's a drag. Automatic breathing, and that you're just sort of going along there breathing now and then, and we do that pretty aptly as opposed to ineptly. And what you don't have to do is stop and say, well, this would be a good time to expand my diaphragm or whatever it is you do there, but you've got a lesion in this part of the brain and you lose the capacity for automatic breathing. Okay, somebody who's not in BioCore, tell me, what do you die of if you have Odine's curse? Oh, who just mumbled that? Asphyxiation. Good. Okay. And at what particular time? Yeah. Or you actually don't die of asphyxiation when you're falling asleep. How come? Because you keep waking up. You fall asleep, and 30 seconds later you wake up because you're on the edge of asphyxiation. You die of sleep deprivation. How's that for one creepy thing to have happen to you? So you can start obsessing over that as an eventual problem. Here's the boring part of the brain, this reptilian stuff. We do this part of the brain exactly the same way that lizards do. Sitting on top of it is the area that we now put all of our attention and love upon, which is the limbic system, this emotional part of the brain, which we will soon see in sickening detail. The limbic system is mostly a mammalian invention. Birds, reptiles, fish do not have especially complex emotional lives. Uh-oh, there's people from Russ Fernald's lab in here. Do not tell him I just said that. But yeah, not a word about it. Okay, but it is mostly a mammalian invention, greatly expanding the limbic system, emotional complexity. Stay tuned. The next hour and a half is about what is much more subtle than saying, ooh, this does emotions for you. Sitting on top is the cortex, the gleaming analytical machine of cortical function and cognitive expertise that we have. This is something that all sorts of species out there, fish and birds and not bees, but birds and almost bees. And what you got there is one that nonetheless is greatly expanded in vertebrates and mammals, and greatly expanded even more so in primates, and even more so than us. And as we will see, one area of the cortex, which is really very uniquely human and very intertwined with the limbic system. As we'll see, what is critical in making sense of the cortex with respect to all this stuff, this limbic emotion behavior stuff, and everything coming for the rest of the course, is the rumors of the cortex being this rational, independent, analytical part of the brain that goes about its business while all this hormonal, emotional muck is going on south of it is completely wrong. All sorts of aspects of cortical function are being influenced by hormones, and thus, indirectly, under the regulation of here, All sorts of aspects of cortical function are being influenced by the limbic system, and all you have to do to appreciate that is think back on times of extreme emotional duress and arousal where you made some incredibly stupid decision that seemed like a brilliant idea at the time. Therein is proof that your limbic system is able to influence decision making going on up in the cortex. And as we will see, of great relevance to us as a fancy species, the cortex has vast ability to influence stuff going on below there. And all you need to do to prove that is sit and think about the fact that you are not going to live forever. And suddenly this part of the brain is going to be kicking out CRH and things like that. And you've just done it with thought. There's bidirectionality that completely does in the notion that here's the emotional part of the brain, here's the cognitive part, and that's completely wrong, that they're separable. Major figure in the field, a guy named Antonio Damasio, who's probably the best thinker about any of this stuff, had a book a number of years ago called Descartes' Error. Descartes, who saw that there was this great potential separation between thought and emotion, and that's complete nonsense. So starting off merely on an anatomical level, which is to say which neurons where are sending axonal projections where else, tons and tons of crosstalk between the limbic system and the cortex. Okay, so we now have this utterly undefined mass of limbic system, and we also have a tabula rasa there, but here it is. Okay, but what you've got is, of course, subparts of the limbic system. Subparts out the wazoo. There are people who spend their entire lives arguing about whether this subnuclei of this part of the brain should logically be considered as having 7 subparts or 8 subparts. Extremely complicated part of the brain. And what we'll be looking at here is the circuitry. The circuitry and what each individual subregion of the limbic system does. All of this got sorted out in the 1930s or so by a neurologist named James Papes, and I think there remains enormous controversy over whether his name is pronounced Papes or Papez, but probably neither is correct. But what you wound up getting as a result of his work was the first person to begin to think in a systematic way of, hey, all of these structures in that old rhinencephalon part of the brain, all of these structures are sending projections to each other. This seems to be some organized module of interconnected function, and it was around that time that people began to be seeing what some of the effects were, what some of the roles of these were behavior, and suddenly out comes this is the emotional part of the brain. And the circuitry that's since been delineated is known as the Papes circuit or the Papez circuit or the whatever the guy's name was circuit, and we will see that shortly. He was the first person to begin to see, just on the level of connections, that this is an area that seems to be doing a lot of interrelated functions. Quick definition, what do we mean by doing connections? Again, back to last week's introductory stuff, a connection, a projection, neurons in this part of the brain are sending their axons to that part of the brain and forming synapses there. This area projects to that area. So where are the projections, where are the connections to translate this neuroanatomy concept into sort of the most accessible level? What parts of the brain are talking to this region? And what parts of the brain is this region talking to? Where are the axons coming from? And where are these neurons' axons heading off to? So circuitry in that regard. So this is going to be an enormously complicated circuit, but at the end of the day, there is one way to think about it, which is unifying, which clarifies everything, which is the thing that every single nucleus and subnucleus and subsubnucleus in the limbic system, the thing that everything in the limbic system wants to do is tell the hypothalamus what to do. The entire limbic system is structured around trying to influence hypothalamic function. There's all those connections going to the cortex and other areas as well, but for a first pass, the way to conceptualize the limbic system is it tries to influence what the hypothalamus is up to. Why would that be interesting? You know that from last week. Hypothalamus is the central hub for all of that neuroendocrine stuff, and thus the entire world of your brain wanting to influence behavior and emotion in any sort of endocrine context, you're going to have to be talking down here. From earlier last week, hypothalamus also playing a pivotal role in the autonomic nervous system coming down the spine, the whole world in which emotions change how your body works is all about the limbic system telling the hypothalamus what to do. And thus what you see is all the circuitry in here is built around trying to influence hypothalamic function in a certain way as follows, which will make perfect sense, which is what every single area of the limbic system wants to do is tell the hypothalamus what to do. And in addition, the other thing that it really wants to do is make sure no other part of the limbic system is telling the hypothalamus what to do. And thus you've got all sorts of areas with means of stimulating regions of the hypothalamus and inhibiting the activity of other limbic regions. So you've got all these patterns here of stimulating down there, inhibiting here, all built around these different regions wanting to influence hypothalamic function. So amid it being a total, complete mess of circuitry, one rule helps you begin to sort out the details, which is, okay, so you've got this area of the limbic system, and it wants to influence the hypothalamus at the end of the day. How many synapses away is the hypothalamus? How many neurons are intervening between this part of the limbic system having something to say, getting action potentials, and influencing something in the hypothalamus? This winds up being a pretty good rule of thumb in terms of looking at influence. You send a projection straight into the hypothalamus. In other words, you are one synapse away, and you're going to be having a lot more control over what's going on there than if you have to send a projection here, which sends one there and there and bouncing around, and 14 synapses later, you're influencing hypothalamic function. How come? Well, for one thing, one synapse away, you're going to be a lot faster. And the other reason being, if there's 14 synapses in between, everybody downstream of you is going to have their own opinion about things, and it turns into a game of telephone. The most direct way of influencing hypothalamic function is to have as few synapses in your projection as possible. And thus what you see is most limbic regions have a number of different ways of getting information down to the hypothalamus, and they differ in the number of synapses, and thus they differ in their power. And what we'll see is just one example here. We'll do that later. Okay, so lots of examples of that. Just to give you a sense about something interesting in us in terms of this rule of how many synapses away. So we've got this limbic part of the brain, and back to that rhinencephalon business. That, rodents are super olfactory, and fish do whatever they do, and again, don't tell Fernald, and all these other species have their, and we're not a particularly olfactory species. Here's something interesting. Every single one of your sensory modalities has to go through a minimum of 3 or 4 synapses to first plunk down any influence in the limbic system, except for our olfactory systems, which are 1 synapse away from the limbic system. So even us, we are not terribly olfactory, and we've got atrophied olfactory bulbs. That's a rat. Our olfactory system takes up less than 5% of our brain. Nonetheless, olfaction is 1 synapse away from the limbic system, telling you something about that whole world. That visual information can tell you a lot about calculus equations. Olfaction is the thing that flashes us back to some emotional setting. That's the reason for it. Great example of this, counting the number of synapses strategies. The fewer synapses, the more influence. So, what are the structures of the limbic system? First pass, unfortunately, it is going to make sense to learn all the details of these different subregions, because you will be hearing about all of them again. You won't be hearing about them Monday night, but you'll be hearing about them all in the next month or so, and so learn this stuff. Okay, so what's going on in the limbic system? What is inside here? What we've got are the major structures of the limbic system. Sitting in there, you recognize the pituitary, and thus sitting right on top of that is the hypothalamus. And here we have all these other regions. First off, the amygdala. The amygdala, I have already mentioned it a number of times in class. We will hear details soon about what it does. You've already heard some intimations of it. So the amygdala is this cluster of neurons beginning to get another anatomical concept here. You've got nuclei, clusters of neuronal cell bodies, and you have projections, areas of lots of axons. So you will have a whole lot of cell bodies here, and sending their projections off there, you will wind up seeing this as a densely packed area of cell bodies. Each one of these areas I will be mentioning now are areas where the cell bodies are, nuclei. Interesting elaboration, as you also learned last week. One of the things that makes action potentials go really fast is that business about myelin, myelination. These are very membranous wrappings around, membrane, very fatty membrane lipid stuff, which happens to be white in color. Cell bodies tend to be gray in color. And this is where the dichotomy comes in brain science between gray matter and white matter parts of the brain. Gray matter are nuclei, cell bodies packed in there. White matter are cables of axons going from one place to another, wrapped in myelin. So just translating that into sort of terms that we're familiar with. Okay, so starting off, the first nucleus of the limbic system, the amygdala. The amygdala, if you study neuroanatomy, most of these places come with some terms derived from Latin or Greek or something or other. And the amygdala means almond. It means almond. A number of years ago, I reported that it means peach pit, and that turned out to be wrong. And I was also wrong as to whether it was Greek or Latin. I probably should have written that down from 2 years ago. But from some stupid old classic language, this means almond, and it looks like an almond. Okay, next we have in front of it the hippocampus. Hippocampus, which we will hear lots about as well. And you will see the hippocampus is a big structure spanning over there, relevant to its function. Hippocampus, Latin for seahorse. But if you look at the hippocampus, it doesn't look at all like a seahorse, suggesting neuroanatomists don't go out to the beach anywhere near as often as would be a good idea. It actually looks like a jelly roll. But people who spoke Latin didn't know about jelly rolls, and thus they had to see horses. So you got the hippocampus here. We will shortly be seeing how these two communicate with each other, where the axons are going between these nuclei. Next, an area called the septum. And there's a number of septums in the body. Septum is a term for a midline structure. You have a septum separating the two nostrily parts of your nose. The center of the four chambers of your heart is a septum. Septum here is a structure that's right at the midbrain, at the midline of the brain. Then we have, just tucked behind the hippocampus, something called the mammillary bodies. Yeah, you got to know these terms. You will eventually be so grateful for having all this stupid terminology under your belt. It will make the rest of your life far richer. Okay, so you got mammillary body here. Sitting on top of that is a remarkable structure that you probably could have intuited was there because you know about the hypothalamus. Hypothalamus, what does that mean? It means it's the thing that's under the thalamus. I bet you there's something a little bit north of there called the thalamus, and here it is, which is possibly also known as the supra-hypothalamus, but instead it's known as the thalamus, and we will see some stuff that it does. Next are two very interesting areas, which we will hear tons about in lectures to come, in particular the lectures on depression, a region called the ventral tegmental area, and a region called the nucleus accumbens. Every one of these is in the handouts. Don't freak out about trying to get down the terms at this point, just at least get the abbreviations. Here are the major players in the limbic system, with one addition. And this is one that bedeviled the field for a long time and reflected a very, very great piece of insight. A neuroanatomist back who sort of was in his prime in the latter part of the last century, a guy named Wally Nauta, a Dutch guy who was arguably the best neuroanatomist of sort of the last half of the 20th century. And in the 1950s, studying neural connectiveness, what he came up with was a totally nutty idea. Because at that point, we were about 10, 20 years into the limbic concept. These are subcortical areas involved in emotion, and they all send projections to each other. And Nauta, based on looking at the anatomy, what parts of the brain were sending projections where, what parts were getting projections from where, Nauta said one part of the cortex should actually be classified as part of the limbic system. Oh, this was heresy. He was driven out of the neuroanatomy club, and he was insisting that this made sense anatomically and predicted it would make sense eventually functionally, and he was totally right. The most interesting area of the cortex, a region called the frontal cortex, also known as the prefrontal cortex. An obviously real distinction there. We will ignore that distinction. An area, the frontal cortex, including a sub-area called the anterior cingulate. This is a part of the cortex which is intimately interconnected with all these subcortical regions. It is arguably the cortical component of the limbic system. Why is that the case? We will see amid lots of cortex doing stuff like telling you, is it here that you were just stimulated, or was it there? Did somebody just play an A or an A flat? Which limb do I want to move right now? All sorts of nuts and bolts stuff. How do I do long division? All those things. The frontal cortex has this world of utterly uncortex-like things that it's interested in: emotional regulation, impulse control, long-term planning, gratification postponement. It is without question the most interesting part of the brain. And I say that as someone who's just spent like the last 30 years of my life just obsessing over the hippocampus, and I was wrong. The hippocampus has done well by me. I have been faithful to it, but nonetheless, it has struck me more and more that this is where I should have been sitting, because this is, so much more interesting part of the brain than some dumb old area that remembers stuff for you. Frontal cortex is what makes us most definedly human. It is proportionately larger in humans than in any other species. It is the most recently evolved. Something that we will hear about in coming weeks is it's the last part of the brain to fully mature. Most of you in here do not yet have a frontal cortex, which is fully online, which is an amazing thing. Frontal cortex isn't fully myelinated until in your mid-20s on the average. This is not, you were born with all these neural connections. This is a part of the brain that does impulse control for you, and it is not working yet very well in most of you guys for a while to come. I don't know what this explains, but it no doubt explains a great deal. This is an incredibly interesting part of the brain which has nothing to do with, let's turn that sensory information into a three-dimensional map and I could recognize whose face it is. And this instead is a cortical area utterly intertwined with what's going on in the limbic system. One interesting measure of it, which is looking at this frontal part of the cortical brain, and an interesting measure worked on by a guy named Ian Dunbar some years ago, looking at, by now, about 150 different primate species, and in each one asking what percentage of the brain is made up by this prefrontal cortex, how much of the brain is devoted to it. And what he showed across 150 different primate species, the single best predictor of how big the frontal cortex was going to be was how large is the average social group for this species. In other words, you sitting around in some species where you've got to keep track of 3 other people in there, or you live in a species where there's 150 individuals in your troop, and the latter species is going to have proportionally a far bigger frontal cortex. What this suggests on a certain level is this utterly defining part of the brain evolved for gossip. and social relations and appropriate behavior and social intelligence and all sorts of stuff like that. Truly interesting part of the brain, utterly intertwined with the traditional limbic region. So this Nauta guy was completely correct. Okay. So now adding in the next layer of miserable factoids you need to have, which will show sort of the same principles I've been talking about, which is how these areas connect with each other. At this point, it has basically been shown that every limbic region projects to, sends axons to, some other limbic region. I'm just going to put in some of the main pathways here. First one, called the amygdalofugal pathway, which carries information back and forth between the amygdala and hippocampus. Amygdaloid neurons projecting to the hippocampus, hippocampal neurons projecting back. And as we will see, an awful lot of what the amygdala is involved with, I think I've mentioned already, fear, anxiety, learning to be afraid of particular stimuli. A lot of what the hippocampus does is help you remember stuff. This connection between them is critical when you are learning that every time the Russian guy with the beard puts the light on, you're going to get a shock and then you're going to have to salivate when he tells you there's food coming. What the amygdala is doing there is hijacking the hippocampus at that time to have hippocampus do some memory formation for you about a fearful stimulus. So interconnections there that are extremely important. Next, we've got the hippocampus needing to talk to the septum. And it does so with a series of projections, bidirectional again, called the fimbria fornix. Fornix, here's showing just how much fun neuroanatomy can be. Fornix is from Latin or Greek or whatever for arch. And apparently it comes from referring to the Roman Colosseum, and supposedly this is the origins of another word that during classical Roman times, this is where prostitutes would be. So people who would say they were going off there would be going off to the arches, off to fornicate. That's the origins of that. So isn't this such a cool subject? So here we got the fornix there, completing this arch, hippocampus talking to septum. Septum then sending a huge projection coming down to the hypothalamus, back onto the mammillary bodies, and projections going back this way as well. A major limbic pathway called the medial forebrain bundle. Okay, just to make things more complicated now, we now have another projection from the amygdala that goes straight to the hypothalamus. And this is a total pain. This is a pathway called the striae terminalis. And by all logic, what it should do is go from here to there. No, that's not what it does. Here's what the striae terminalis does. It starts in the amygdala, and it goes looping just on the outside of the hippocampus around this way and lands back there. What's up with that? That is ridiculous. What you see here is a principle about a lot of this neurobiological wiring stuff, which is you get totally nutty, inefficient, going out through left field projections. How did that happen? What that tells you is at some point embryologically, these two structures were not so close to each other. And part of embryonic development involved flipping over like this, and suddenly the amygdala is right next door long after it made the most efficient possible connection by way of this, and now you got this completely nutty way to get around there. So often you see really inefficient pieces of wiring here telling you something about embryonic development. Some of the time, you also see nutty pieces of wiring telling you something about the evolutionary history of this nervous system. Here's one example, and this is one outside of the limbic system, but great demonstration of this having to do with some motor systems in the brain. Okay, you're here, and you decide what you would like to do is bend this one finger. What would be the logical thing? Some part of the cortex up here involved in motor control that we're not interested in should send a message down to this finger saying, bend. No, that's not what happens. One part of this motor system called the pyramidal nervous system sends a message to all 5 of your fingers saying, do this. Meanwhile, there's another motor system called the extrapyramidal system that quickly sends down a signal and says, if you happen to hear the pyramidal system tell fingers 1, 3, 4, and 5 to bend, don't pay attention to it. and suddenly you got this going on. Like, what is this? A committee that came up with this? This is totally crazy, inefficient wiring until you think about the evolution of the system. As follows, there's not a whole lot of species out there that have a need in terms of reproductive success to play trills on the piano. There's not a whole lot of species that have to do independent movement of fingers. The basic wiring of this motor system is one of this crude pyramidal system of just sending messages down the line there, extend claws, retract, all that sort of thing. And along come primates and raccoons and a few other things that suddenly get in their heads to move fingers independently. So you got a choice at that point. You can either renovate the entire pyramidal system and rip it out and rewire so that each finger is controlled independently, and that's never going to work because the contractor doesn't show up and it's been a million years. Or what you do is you have to come up with a second, newer system that's superimposed on top. Totally ass-backwards, inefficient way of it. What does that tell you? The extrapyramidal system is more recently evolved. It is more of a primate system. And that's where you get the fine control. What you often see is bits of really clunky wiring and regulation. Tells you things about embryonic life, and it tells you about evolution. Back to this classic quote, which is, evolution is not an inventor, evolution is a tinkerer. It is just playing with what's already there and trying to come up with something better. That was the idea from lectures ago, which all things considered, squid do not swim anywhere near as fast as barracuda, but for something that used to be a barnacle, they're pretty good swimmers. You have to realize where things started and where the adaptations came from. Okay, so more wiring. And now we've got the mammillary bodies talking to the thalamus, the mammillothalamic tract. And then you've got the thalamus talking bidirectionally with the frontal cortex. So you begin to see the starts here of kind of a looping circuit and going bidirectionally, all of that. Two additional areas, this ventral tegmental area and this nucleus accumbens. Ventral tegmentum sends a lot of information here, and from on there it goes to every single place, and we will eventually see great relevance of that bidirectionally. Okay, so horrible circuitry. What you should already see is one example of what I was telling you about before, counting number of synapses. So you're the amygdala, and you want to tell the hypothalamus what to do. You got a choice. You can do it by way of this striae terminalis thing, which is one synapse away, or you could do it by talking to the hippocampus, which talks to the septum, which talks to the hypothalamus. Where's the most important stuff coming out of the amygdala to the hypothalamus? By way of this striae terminalis. What's this communication about? A lot of the time it's the amygdala trying to get the hippocampus to shut up so the amygdala can dominate hypothalamic events. That begins to show this principle of how many synapses away. So we have all of this incredible wiring here, and again, every single one of these, there's other minor ones there as well, and they all interconnect with each other, but these are some of the major ones. Does this make sense? How about now? Okay, so that's the limbic system in terms of basic wiring. So what everyone needs to do now is spend the next 5 minutes filling in what those abbreviations were for, and then we will finally get past this stupid A sends to B and begin to look at function. Okay, so 5 minutes. Okay, so pushing on. Good question that just came up, relevant to biocore people. So what I just said, that business about the olfactory system is one synapse away from the limbic system. Isn't it totally amazing? Remember biocore stuff, that business, the visual cortex, the first layer that does dots, the second layer does lines at certain angles, all that's where you're getting all the synapses coming in, which delay you from getting information into there. Auditory system has the same multi-layer processing. Tactile, all of those are like that, except for olfaction. That drops its first signaling right off into the amygdala. So very, very different circuitry. Okay, so now finally getting to function. So how do you figure out the function of the limbic system? A bunch of different techniques, bunch of different experimental techniques. First, you have the greatest thing that has been most helpful in all of the history of neuroscience in understanding how different parts of the brain work, which is endless warfare. Because what that gives you are people who have had parts of their brain blown out of the water there. You have, nice jargon, missile wounds. So after every big, good, fun, energetic war, all sorts of neuroanatomists get their careers like jump-started for years afterward, studying people who have had parts of the brain lesioned, damaged, destroyed by circumstances like that. But, you know, any old-time neuroanatomist are delighted to get a hold of somebody who's had a brain region damage. There's one guy, in fact, everybody has heard of HM, I suspect. Anybody not heard of HM? Okay, so you've all heard of HM. There's another guy, NA. NA has his septum taken out, and what happened to NA? All that it ever says in the literature was that he was at a wedding and his septum was destroyed by a miniature fencing foil. That's it. That's the literature on what happened to NA. So NA, you know, was a draft dodger in endless different wars, and then he gets done in by his like sister-in-law's wedding kind of thing. But lesions are critical. So human studies where you have had accidental damage to brain regions, or the rare, horrifying world where there was damage done to human brains intentionally, back to class 1, the frontal lobotomy business. Just to clarify, frontal lobotomies in its classic form was not lesioning the prefrontal cortex. It was just putting a big old cut somewhere through there that took out limbic structures, given the fact that most frontal lobotomies were being done, I kid you not, with an ice axe. That was the tool of choice for the neurosurgeons. This was not a very precise surgical technique, but it was taking out all sorts of limbic structures. So the rare world of intentional lesions in humans, HM being the rare version of that where it was done for a valid clinical reason. Much more often in the lesion world is taking experimental animals where you go in and you selectively destroy some region. And the strategy, whether in humans or animals, is absolutely obvious, which is, okay, if we destroy this part of the brain, what doesn't work right anymore? Ooh, we've just found out something about what that part of the brain does. Next strategy, classic one. Now, instead of lesioning a brain region, you put down an electrode where you stimulate that region. You artificially generate action potentials there. This is a brain region that gets projections from here. And this region is not saying anything whatsoever. There are no activity here to send action potentials. Nothing's happening. And what you're doing instead is by sticking an electrode in there and stimulating, you are simulating an input there. Stimulate and now see what happens. Ooh, we've just learned something about that part of the brain. Rarely done with humans, except for epileptics with severe types of intractable epilepsy, where a certain type of neurosurgery is done to correct it, and it's often quite effective. On the way down, the electrodes going down, often the neurosurgeons stimulate on the way down to be assured that they're in the right place. Okay, next version. Now instead, you put down an electrode, and that one's a recording electrode. What that one is telling you is, so you're down in here, when are these neurons getting electrically excited? Or putting it in here, when are action potentials pulsing through this part of the brain, picking up what's going on? And what really winds up being fancy are people who, for example, can put a stimulating electrode down here and a recording electrode there, that sort of thing. There's a whole world of incredibly adept people who could record from one single neuron at a time. Or how's about this? Back to those neurotransmitter receptors coupled to an ion channel that opens up and stuff goes in or out. There's a technique called patch clamping where people can record from one single ion channel at a time, which is just madness that people could do this. But in terms of the resolution, all of these techniques artificially get neurons excited electrically, see what happens next, put in a recording device to tell you what's happening, and thus you begin to do things like, ooh, every time I do this with my hand, these neurons get action potentials. We've just learned something pertinent there. What else? Another realm of techniques you've already heard about, which is this plain old boring anatomy. Which part of the brain sends axons to this part of the brain? How many synapses away? All of the means of deriving information that we heard about before. So that's the whole world of just looking at the circuitry, telling you about function from that. Then, of course, there's a whole world of biochemical stuff. You are measuring levels of things. You are measuring levels of neurotransmitters, molecular biology, seeing where different genes are being expressed. If you see genes that are only expressed in this part of the limbic system, under some circumstance, you've just learned something about its function, all that sort of thing. Finally, a world where you could do really interesting, trendy stuff with humans in terms of seeing what's up in the limbic system, where what you do is you can image brain regions. And this is now a whole world of, instead of sitting around waiting for somebody to die and slice their brain up afterward, instead you could do imaging, CAT scans, CT scans, MRI, where you see the areas of different parts of the brain, how big or small they are. You can look at the metabolic rate in certain areas of the brain, which is sort of the equivalent of recording. Stick in an electrode and you can tell what's happening here by when there's electrical excitation. Use functional brain imaging and you can tell what's happening here by when suddenly there's more oxygen or glucose being consumed here. Same sort of information. With the imaging though, you're getting a picture of the entire brain all at once. And one of the cool things that's come out of those studies is showing that some regions of the brain, particularly the limbic system, will change their size over time. One example, which I think we've heard about already, which is that the amygdala gets bigger in people with post-traumatic stress disorder. Severe traumatic history, and this part of the brain expands. People with PTSD, amygdalas are larger. You look at the metabolic rate in the amygdala, people with PTSD, it is more metabolically reactive than other areas of the brain, or than in people who don't have PTSD. So that's just told you something interesting. Nice cellular basis of that. Periods of severe stress cause neurons in the amygdala to grow more dendritic processes. That's probably why the amygdala gets bigger. So that's the sort of thing that gets picked up on humans with brain imaging. Another version over in the hippocampus, just as pertinent to some of the stuff we will hear about, people with long-term major depression, their hippocampus gets smaller. Their hippocampus atrophies a bit. And the same sort of information being picked up, you see an area of the brain that is plastic enough, that is malleable enough that its size will change in response to certain types of experiences or emotional states. You just learn something about what that part of the brain does. So that winds up being extremely informative when there's differences in the size of different brain regions in response to experience. Okay, so that's generally the set of tools that are available to people trying to figure out what's going on in the limbic system. And all of these tools come with some important limitations. These are limitations if what you're trying to do is figure out what this part of the spinal column does instead of that part, because you need to figure out all sorts of caveats in there. If that's hard, it is a gazillion times harder to use some of these techniques to make sense of emotion and emotional memories and things of that sort. What are some of the difficulties in interpreting data from all the results we just heard about? Here is one problem. Okay. Every day, unbeknownst to you guys, there are 2 to 3 dozen huge tractor trailer trucks that start at dawn from Gilroy and bring us our garlic that is used in all of the dorm dining rooms. Every single morning that happens, thousands of pounds that are consumed in the dorm, and you are trying to understand where in California is the garlic generating center, the garlic nucleus, and what you've been selectively doing is carpet bombing certain areas in Northern California, and as a result of a direct hit on 101, no more garlic shows up at Leland Stanford Junior University. So what have you just concluded? 101, somewhere right around Laser Quest, is exactly where the center is for producing garlic in Northern California. And that's the only plausible scientific conclusion you can reach. What we have here is the problem of centers versus fibers of passage. Have you just, in a lesion study, found out something about a region, you've destroyed it, and something doesn't work normally? Is that because you've taken out the neurons, the cell bodies in a true center, or have you inadvertently cut a fiber passage? Have you blown out 101 and endless mistakes made over the years early on in neuroanatomy, people failing to distinguish between lesions of nuclei versus lesions of pathways. What really winds up being hard is when you have nuclei with cell bodies and at the same time fibers passing through from someplace else. That's what this medial forebrain bundle is about. Cell nuclei all over the place, but at the same time fibers passing through. When you do a lesion in a place like that, you can't tell whether it's the cell bodies, the nucleus aspects, the center that you're taking out, or whether you have just taken out a fiber of passage. Enormously difficult stuff to do. Okay, so that's one constraint. Once you get through that and you figured out, ooh, this really is a center, this really is the cell bodies rather than just the axons, then you deal with this issue of what does a center mean in the brain? Yeah, this may be the center, the part of the brain that moves this finger instead of this one. That may be a pertinent concept. Ooh, this is the center of the brain for feeling depressed amid a bittersweet gratitude for the unexpected pleasures of life, which are felt in a poignant context, making you smell memories of the kindergarten classroom you were in. Oh, that's the center of the limbic system for that. By the time you're getting to a part of the brain that's doing all this emotional stuff, intermixed with sensory information, intermixed with memories, even the concept of, oh, this is the part of the brain that does fear for you. This is the part of the brain that does aggression. So we'll see, these are some pretty flimsy concepts. Okay, more qualifiers. The next thing you need to be in order to be a good neuroanatomist and make use of the information from these techniques is to also be an ethologist. We already heard a version of that, the rhinencephalon. That's what's going on in a species that's all about olfaction. You can't understand the wiring going to the limbic system unless you have interviewed an animal in its own language. Classic need for ethology. Okay, so here would be another demonstration. You have found a part of the brain which you think is interesting. It's a limbic structure. Nobody quite knows what it does. And you have now just stuck a stimulating electrode down in there. And the strategy is you're going to mildly stimulate there and you see what happens in the organism. So your first study subject is a lion, and you've stuck your stimulating electrode down in there, and you mildly stimulate this part of the brain, and what the lion does is it does this. It extends its claws. Now, your next subject is a human. So you sit down, you plunk down your stimulating electrode, and you stimulate mildly in that same part of the brain, and the human says, shit. So, what part of the brain have we just found here? We have found a part of the brain that plays a role in expressing a certain degree of irritation. And if you're a lion, you're sort of extending your claws there. You probably want to stop messing with me because look what I'm doing right now and look how sharp they are. With the human, you gotta know your species. Another example of this. So here's a part of the brain, and you're wondering what's up with this. You do the same strategy. You stick in the stimulating electrodes, and you do this to a rat, and what she does is proceed to run around her cage like crazy and take pieces of newspaper, rip them into little pieces, and stuff them in the corner. Okay, that's an interesting center of the brain. Then you do the exact same thing to a rhesus monkey, and what she does is grab any sort of cylindrical object around and hold it in her arms by her nipples. Oh, maternal behavior. First case, you got a rat making a nest. Second case, you were stimulating nursing behavior. Ah, okay, you got to translate it into what it looks like in the species. And here's one domain where that went utterly, utterly wrong. Part of the hypothalamus, which people used to think had everything to do with aggression. Because, for example, here you would take a stimulating electrode in a rat, and you put a mouse in the same cage as the rat, and you stimulate that part of the brain of the rat, and the rat leaps on the mouse trying to rip it to shreds. Now you take a human and you stimulate that same part of the brain, and they leap up and run over to the cereal box and rip it open and start eating. What's up with that? Oh, rats eat mice. That's what that was not a rat being angry, that was a rat getting something to snack on. And you had to recognize the difference between what does aggression look like in that species versus what does food acquisition, and if it's a predatory species, it could pass for looking mighty aggressive and not actually be. A whole generation of people thinking they were understanding the neurobiology of aggression inadvertently made this mistake and wound up studying the neurobiology of predatory behavior. So you gotta know your species ethological principles. Next, you gotta know your individual. So here, now you've got two lions, and you stick in your stimulating electrode into the first one, that same region again, and you stimulate, and he does the same deal again. Or suppose you even stimulate it more, and he does this a bunch of times, and then he roars savagely. Meanwhile, you stimulate the same area in the second lion, and nothing happens at all. You take two baboons, you stimulate that region in the first one, and he gives this big threat yawn displaying his canines. You stimulate it in the second individual, and nothing changes in his behavior. What's the difference? Any speculations? Dominance. Yes, dominance. Here, in the second case, in the first case, you're doing it to a dominant individual, and they do their fixed action pattern for their species of expressing aggression, dominance, whatever, but you do it to some subordinate guy, and that's not part of his repertoire right now. And even though that's the part of the brain that does that behavior, it is tightly inhibited in that individual, because that's not an individual who goes around displaying his canines to a whole lot of his buddies. You have to know the individual as well. So given all of those constraints, that tells you if you're going to understand what's up with this part of the brain, the difference between fibers and nuclei, and don't get overly impressed with the notion of centers of function, but you got to do the ethology stuff. Here, more than any time that we've seen, you got to understand the species and the individual that you are interviewing with your electrodes. Okay, so given all of that, what do some of the limbic system structures do? This is a first insanely simplifying pass, because every one of these areas you're going to hear about so much more, but a very first pass, and you notice as part of the first pass, I'm going to be violating the caveat that I just gave 2 minutes ago, in that on this first pass here, I'm going to be presenting these as the center. This is the part of the brain that does this. We're going to see how totally wrong that is, nonetheless on a first pass. Amygdala. Amygdala is centrally involved in fear and anxiety. You know this already. Of tremendous significance, telling us something about why this world is such a messed up place, the amygdala also plays a central role in aggression. Not messed up just because, this is the part of the brain that generates aggression, but what it tells you is you cannot understand the neurobiology of being violent without understanding the neurobiology of being afraid and being anxious. And the fact that this is the same part of the brain that does both of those functions, suggesting on a certain neurobiological level, in a world in which no neurons need be afraid, you're not going to be generating a whole lot of aggression. Also, very interestingly, what we will hear about is the amygdala also plays a role in male sexual motivation. What's that doing going through the amygdala? Not sure whatsoever, but what that may begin to explain is the subset of individuals who will confuse aspects of aggressive behavior and sexual behavior in all sorts of pathological ways. First pass at what the amygdala is about. Meanwhile, next door, we've got, not next door, but 2 synapses away, we've got the septum, showing a first important example of this theme in the limbic system. Amygdala mediates aggression. Caveats, all of that, we got the fine print now. The septum inhibits aggression. It does exactly the opposite, beginning to present a theme throughout the limbic system of different sub-areas working in opposition. What would you be logically betting the farm on right now? When the amygdala is activated, it tries to silence the septum. When the septum is activated, it tries to silence the amygdala, cross-inhibitory projections. So what would these things look like? You destroy the amygdala of an animal, you never get aggressive behavior again. You record from it when it's smelling some scary rival. The amygdala activates, you stimulate, and it does an aggressive behavior. You could begin to see how you figure this out. Next, hippocampus. Hippocampus, famous for being involved in learning and memory. Important for us as well, hippocampus plays a role in turning off the stress response. You remember that negative feedback stuff from 2 days ago. Every endocrine system, there's got to be a part of the brain that measures the hormone in the bloodstream to figure out what should the hypothalamus be doing. The hippocampus measures the levels of glucocorticoids. Why should a part of the brain that does memory be so intertwined with stress? Very logical in 2 ways. Number one, here's a stressful, horrifying, scary circumstance. Remember what you did to get out of this if you do manage to get out of this. File this away very, very emphatically. Second thing that it's good for is stressful circumstance coming up. What did I do last time to get out of this circumstance? So an intertwining between a memory part of the brain and a relationship with stress hormone regulation. Next, mammillary bodies has aspects relevant to maternal behavior. And that's not why it was called the mammillary bodies, but they're shaped by like mammary glands, if you, once again, don't spend a whole lot of time or look too much at seahorses, but the mammillary bodies. Next, prefrontal cortex, you've heard all about, and we are going to hear tons more about it because it is incredibly interesting in terms of maturation and learning appropriate sexual behavior, learning not how to be aggressive, but when to be aggressive. The notion that there is a problem with this part, the anterior cingulate, in people with clinical depression. Anterior cingulate, I think I may have referred to this already. Sit somebody down, poke their finger with a needle, and areas here are going to light up, and spinal pathways telling you it was this finger and not that one, and areas making you breathe faster and all of that. And in addition, the anterior cingulate is going to activate. Take somebody now and have them watch their loved one get their finger poked with a needle, and nothing's happening down here, but the anterior cingulate activates. It's doing something along the lines of empathy and feeling somebody else's pain, and suddenly it becomes real interesting that there's stuff going wrong in it in clinical depression. Easily translated into that is a disease of pathological hypersensitivity to the pains of life and the world. Okay, so frontal cortex. Finally, lurking around in here, the ventral tegmental area and the nucleus accumbens, really pertinent to depression as well. This is the part of the brain that has all the neurons that release dopamine having to do with pleasure. This is the part of the brain that cocaine works on, that indirectly all addictive drugs work on. This is the part, stimulates dopamine release here, stimulates dopamine release at all the interesting places, frontal cortex, amygdala, hippocampus, central to understanding everything about appetitive behavior, behavior driven by an appetite for something. That tells us an elaboration we'll be learning about in terms of what the dopamine system here does. People used to think this is the system that activates when you are feeling pleasure. Turns out it does something much more interesting. This is the part of the brain that activates when you're anticipating feeling pleasure. It's not about getting the reward, it's about anticipating that you will get a reward. And what it's most about is not only anticipating that you may get a reward, but powering the behavior you need to do in order to get the reward. Standard paradigm showing this. You take a monkey that's been trained, every time a light comes on, at that point, if it presses a lever 10 times, 5-second delay, and out comes some food. That reward system. Initially, the very first time it stumbles into just happening to hit this out of boredom 10 times, and out comes some food. Ooh, up go dopamine levels when the food comes out. But pretty soon what you get instead is the dopamine levels go up when the light first comes on. This is not, ooh, I just got some pleasurable food. This is, I know this one. I'm on top of this. I'm all over this one. This one's easy. This is one of those 10 lever press deals. This is going to be great. I got this under control. Elevated dopamine, you go hit the lever, and along comes the food. Now, instead, you flash on the signal telling you we're entering one of those press the lever 10 times, you get food sessions. You turn that on, and you block the rise in dopamine, and the animal doesn't press the lever. What this pathway is about is giving you the energy, the motivation, the depolarization of neurons for carrying out the pursuit of pleasure. And that's really informative, as we'll see, because stimulating the pursuit of pleasure is far more of an addictive process than the pleasure itself in terms of making sense of some of the circuitry. Okay, so all of this eventually plunks us down into the hypothalamus. The hypothalamus has a gazillion sub-areas, all these separate little nuclei that have a variety of functions, all of which have boring, vegetative, just keeping track of body temperature type functions, all of which are also profoundly affected by the whole world of emotion and thought and memory. First area, ventromedial hypothalamus. We're going to hear tons about that starting next week, along with a cognate area, the medial preoptic area. These terms will become familiar. Do not freak out. These areas are very pertinent to sexual behavior. And what we will see is that one of them is more involved in male sexual behavior, one more in female sexual behavior on the average. There are gender differences in the size of these areas. There is one landmark study that everybody learned about 15 years ago, showing that there's a difference in the size of one of these regions depending on your sexual orientation. There is an amazing study that was shown that people who are transgender have differences in the size of some of these regions as a function not of what their sexual phenotype is, but of what they've always said, I have always felt like, or whatever. They have the region the size of the what I always felt like I was rather than what they actually are, bringing in this possibility that what transgender is about is not somebody thinking that they have the wrong sex, but it's somebody who's gotten the wrong body. So these areas we're going to hear lots about next week. Next region, SCN, suprachiasmatic nucleus, and once Julie Andrews says that as loud and fast as possible, the suprachiasmatic nucleus is about circadian rhythms. We're mostly going to ignore that, and anybody who has anything to do with Craig Heller's lab, don't tell him that I said that either, but like stupid circadian rhythms in fish, sort of doing two of my colleagues at once. Next, moving on. Another part of the hypothalamus, PVN, paraventricular nucleus. What's made in there? CRH. That's the part of the brain that initiates the backbone of the stress response. And what areas are sending projections to it? Everything. Everything ranging from pathways that tell you that you've just burned your toe, to pathways telling you you are very, very hungry, to pathways telling you that you have just smelled a terrifying territorial rival, to pathways telling you you have just thought about, oh my God, only 4 days till the midterm or whatever it is. By the time you get to species that can be stressed by very abstract cognitive states, by thinking about the suffering of somebody on the other side of the planet, what do we know about the neuroanatomy? PVN is getting projections from all over the place. Next, arcuate nucleus. Arcuate nucleus is the bottom of the hypothalamic funnel. That's the part where all of the hypothalamic hormones come out and get into the circulatory system there. There's actually another area on top of that. I will not mention it, but that's its function. Lateral hypothalamus has something to do with hunger. And that's the area that people wasted years on thinking they were studying aggression when they were instead studying predatory behavior. Lateral hypothalamus has to do with hunger. Boring version of it. It does things like measure your blood glucose levels and your insulin levels, all sorts of logical things. Really interesting level, evidence that the lateral hypothalamus has something to do with hungers in the broader sense. Hungers for types of information, hungers for other rewards other than food. It's an area having to do with hunger. Okay, so this very, very fast pass here over all of it. One additional component to think about, which is everything I've just said has been oriented around all this limbic system stuff built around eventually influencing the hypothalamus and thus eventually influencing the entire world of hormones or of the autonomic nervous system. This is this unidirectional picture. Stuff starts in the brain and you have outcomes throughout the body. What is just as important is all the ways in which stuff going on in the body will influence limbic function, completing this circle. And an awful lot of that information is the autonomic nervous system not going in this direction, but going in this direction. Now, what this brings up is a classic theory that's been in psychology since around 1900 or so, called the James-Lange theory of emotion, named for William James and Lang, whoever that was. That may have been his puppy. But the James-Lange theory of emotion goes as follows. Here's how you feel an emotion. It is not the case that your brain decides it's feeling an emotion based on sensory information coming in, memory, whatever. The brain decides it's feeling an emotion and tells the body, let's speed up the heart, let's breathe faster, let's sweat, whatever it is. That's not how emotion works. Here's how emotion works. Stimulus comes into your brain, and before you consciously process it, your body is already responding with heart rate, with blood pressure, with pupillary contractions, whatever. How do you figure out what emotion you are feeling? You are getting information back from the periphery telling you what's going on down there. In other words, how do you figure out you're excited? Whoa, if I'm suddenly breathing real fast and my heart's beating fast, it must be because I feel excited now. Ridiculous, totally absurd way in which you could process stuff, completely inefficient, and over the years, after decades of being wildly discredited, more and more evidence that that is some of how emotion is based, your brain being influenced by your bodily state to decide what emotions it's feeling. Number of examples. First one of those hormones that just has emotion written all over it, adrenaline. Adrenaline, epinephrine associated with arousal of the sympathetic nervous system. So what happens when you dump some epinephrine into the nervous system? What does it do to the brain? What does it do to the limbic system? What does it do to the sort of behaviors influenced by it? And greatest demonstration, this was a classic study in the 60s, a guy named Stanley Schachter doing a study that you could not do again on humans with current rules. But here's what he did. He gave people epinephrine, adrenaline, without them knowing it. Okay. Less paperwork in those days, but here's what the one version of the classic experiment was. Somebody comes in and they believe what they're doing is some study of you take this vitamin and does it change the way you do Sudoku or something, and like that was just the teaser, that was the distractor. We've taken this pill which actually contained long-acting epinephrine. Then you're going to go into the study where you're going to get tested or whatever the task is, and please sit down in the waiting room for a while. And the actual experiment took place in the waiting room. Sitting there in the waiting room was another individual, a confederate of Schachter, somebody working on the experiment. In half the cases, what the person sitting there was supposed to do was say, I can't believe it. I was supposed to go in. They said I would have a 1 o'clock appointment for this. It is 1:30 already. I'm going to miss my class. My car is going to get towed. I'm totally pissed off. I can't believe these people are doing it. Meanwhile, half the time, the other person is sitting there saying, God, I love these psych studies. You do a lot of them. I love doing these ones. I had such a cool one last week. Which dorm are you in? And off it goes. And the key thing being totally different emotional states, in each case being exposed to someone in a very emotionally aroused, agitated, expressive state. And what did Schachter show? Does epinephrine make you get more angry? Does epinephrine make you get more friendly? It doesn't do either. It's modulatory. What epinephrine does is it kicks up the fevered level of whatever emotion you were socially being brought into. And thus what you got was the subject sitting there soaked in epinephrine while somebody is like going on about how late it is, and these people, before you know it, they're up there banging on the counter at the receptionist and saying this is an outrage and this is unfair and all of that, getting caught up in it. Meanwhile, the other one, within like 5 minutes, he's singing frat songs with this person and there, and they're going to get together for dinner and they're hugging each other. And what does epinephrine do? It does not cause a behavior. It exaggerates, it magnifies emotional states that have been provoked for other reasons. It modulates the exact same concept as the other day. What does GABA do to that neuron A? It does nothing whatsoever. What GABA does is it modulates the ability of this glutamatergic neuron to talk to A. What does epinephrine do? Does it create any emotions? Not at all. What it does is jack up the intensity of whatever emotions your social circumstances have generated. Another example of James Lang sort of stuff. Okay. So you were sitting around and you were terribly anxious, and you go to a clinician and the person decides to give you, what class of drugs do we know endlessly by now? Benzodiazepines. They give you some Valium to decrease your anxiety. Meanwhile, across town, there's somebody who has a big gymnastics competition on Sunday, whatever, and they've pulled a muscle, they've been having muscle spasms, and this is really a problem. So they go to their clinician, who gives them a muscle relaxant. Benzodiazepines, the exact same dose. So wait a second, here's somebody taking Valium for anxiety, and here's somebody taking Valium to relax their muscles. What's up with that? It's the exact same thing. And it is a very James Lang moment. What's part of anxiety about? It's monitoring the level of tension in your body. It's getting feedback from muscle tone. And that's one of the ways in which anti-anxiety drugs like benzodiazepines work, because you're sitting there and you're saying things are just as horrible as they were an hour ago, but I am so relaxed, I'm like dripping out of this chair here, it must not be so bad. Part of you deciding is monitoring the muscle tone in your body, coming back with autonomic information. That's where you break the cycle. That's why the same drug decreases anxiety and is a muscle relaxant. More examples of this. One is what's going on with meditation. That's what biofeedback taps into. So you're sitting there and you're suffering from high blood pressure. Translated into our terms, something here is driving blood pressure to be increased too much. It's not necessarily that, it could also be blood vessels, but we'll put it on a brain basis here for the moment. So you got a choice between taking drugs or doing some meditational something that you want to hook up to biofeedback to make it more effective. Here's what you do. They sit you down, and they wire you up so that they can monitor your blood pressure. And you sit there and they say, okay, I want you to relax and focus on the best day of your life. Let's see what happens to blood pressure. Nothing much. Okay, focus on your favorite piece of music. Nothing much. Okay, focus on your favorite piece of music that's kind of slow and dreamy. And suddenly the monitor goes this way. The person's blood pressure has just dropped 10 points. And then you say, stop. Wait a second, what did you just think about? Think about it again, and it does it again. And what the person begins to learn at this point is what aspects of meditative states, what aspects of memory, whatever, suddenly allow the brain to be regulating blood pressure. What's going on there? You're using this machine to send the signal up there. Part of what's also going on is feedback that way. So another version of one of these loops. More versions of loops. Here's one that probably accounts for 90% of the arguments between significant others on this planet. Okay, so you and your significant other are behaving, interacting in a primate social biological way, and one of you does something that totally outrages the other one, and they're furious at you, he says, personifying, and they're furious and what's going on, and they convince you readily that you're at fault, and in fact this was a horrible thing you did, and they're all agitated, and you apologize, and you make it sound like you really mean it because you really mean it, and you apologize, and great, and they finally say, okay, I accept your apology, don't do it again, it's all sorted out, it's over with, it's done. And then you begin to think, okay, great, we're out the other end of the woods there, things are just fine now, and suddenly they remember something miserable you did to them in 1968, and they suddenly remember it in every possible detail and want to argue about it all over again. What's going on there? What we have is another James Lang moment when you get into an aroused, angry state, and then it's all over with afterward. That's great because the person has apologized. So cognitively, you have just adjusted things. Yes, they recognize they did the wrong thing. Good, I'm vindicated. That's great. It's settled now. It's perfect. And you could do that in a second. The trouble is it takes a long time. It takes minutes now for your sympathetic nervous system and its consequences to completely coast back to baseline. So you're sitting there and the problem has been solved, but your heart is still racing, and what do you have? A James Lang moment there, sitting there saying, okay, well, I was really upset about that, but that's just sorted out and that's all taken care of, except I'm still feeling totally agitated. It must be for some reason. It's what that person did to me in the second Roosevelt administration. And suddenly out it comes out. It's this James Lang loop there of the physiological information coming back there, prompting one into trying to find a cognitive explanation for it. There must be a reason why I am still hyperventilating. Oh, this must be the reason. This thing we need to go over also. Okay, I'm going to give one factoid here, and you can do with it what you will, but there is a pronounced gender difference in how long it takes after strong sympathetic arousal for things to go back to baseline. Okay, let's just get a show of hands here. Who guesses that it takes longer to get back to baseline in males? Okay, well, unless there's a lot of abstainers here, I think we're, okay, females, yeah. Yes, there is a pronounced gender difference in that, and there's another realm in which you have that same pronounced gender difference, which is after orgasm. Males go back to baseline a lot faster than females do. And that explains a whole world of, like, you know, he wants to go out and get noodles and peanut sauce, and she wants to cuddle and talk and all that sort of thing. And why can't it expect? And it's James Lang's damn fault that that sort of thing goes on. Okay, so we there have another example of these feedback loops. A couple more. Here's an amazing thing. You take somebody with clinical depression and you force them mechanically to take a somatic state that mediates different emotions and signifies other emotions. What am I talking about? You take the person with depression and you say, you are going to mechanically go through the process with your facial muscles of smiling again and again and again, and you force somebody to do that for a half hour or so intermittently, and they feel better afterward. The world is just as depressing as it was 30 minutes before, but there's something going on here saying, if I am getting feedback saying I'm doing this thing with my muscles here and I'm doing it a lot, it must not be so bad. Other studies showing you take somebody and either they are slumped in a chair or you have them sitting up erect and you tell them the score that they got on a test that you just gave them before that, and it's a very good score. And what you see is the more straight upright the person is sitting, afterward the more they will assess themselves as being made happy by the result they got and being made to feel proud. What's up with that? The posture of your spine is doing something up here. What this shows us is, amid this whole orientation, the brain controlling hormones, the brain controlling spinal autonomic stuff, the limbic system telling you all of this outflow from there, completing this big circle back to the very first class, that business of think about something really emotionally laden and suddenly your body works differently. Think about the people who have committed murders in the context of a brain tumor, a period, their hormone levels, their nutritional history here, junk food murders, and all of that, Twinkie defenses. What that is the other half of this loop. Okay. So now you should sit straight up and be proud of yourself because you now have all of the buckets necessary under your belt. So that's the first piece of good news. And that now transitions us to the latter half of the course, which will now be to use all of these approaches in looking at specific behaviors. Here is a sexual behavior. First off, as a good ethologist, do we actually understand what we've just observed for this species, this individual, etc.? What went on in the brain 1 second before that caused that behavior to occur? What's up with the neurobiology? What sensory stimuli caused those neurons to generate the world of ethological releasing stimuli? What hormones in the bloodstream in the last minute, in the last hour, made the individual more or less sensitive to that sensory information, blah, blah, all the way back to understanding the evolutionary legacy of the species of which this individual is a member. That's going to be our strategy in each one of these now, working our way back in time. So there's that great news that you are in that position now to take it on. The other piece of good news is we're not going to do it on Monday. We won't have class on Monday, so you can all get all James Langy and freaked out for the exam Monday night. But quick survey among the TAs. So what do you guys think? Should we have a review session then? Should we show a David Attenborough movie?
Go with Attenborough.
Okay, David Attenborough. So which one are you going to bring in?
We'll watch Life, the BBC version with David Attenborough. I don't think it's really available in the US very much yet, so it's kind of a cool opportunity.
For more, please visit us at stanford.edu.